Okay, hi people. So this specific video is going to be about DNA replication, which is technically a part of 2.2. It's actually part of 2.2.2. Okay, that's so odd. But anyways, that's all I'm going to be looking at because this is kind of complicated. But anyways, let's get into it. DNA replication. So this happens during interphase, which happens during cell division, the cell cycle, whatever. I'll actually talk about the whole thing, but specifically DNA replication. Basically, the way that DNA is structured, it's called a double helix. Basically, what that means is it's two strands that kind of wrap around each other, like that, I guess. And so the way that DNA is set up is one side has, one side has different kind of code to it, and then the other side has a matching code. So blue is always going to be partnered up with red and vice versa. So here, every time there's a red, there's going to be a blue, right? This is just the way that DNA works. These are called nucleotide bases. These are the letters. So A is always going to be next to T, and C is always going to be paired up with G. I hope you can see that. So what this means is that if you have just one side, you can tell what the other side is going to be because of these pairings, if that makes sense. So that's just a little background information. DNA replication. There are a couple of different steps to it, but so we've got our DNA here. This is called the parent DNA. So if you imagine parents multiply and have children, right? This is going to be multiplied. This one parent DNA is going to be multiplied into two strands of DNA. DNA replication. So step number one, a big, I'm going to choose purple, a big enzyme called a helicase enzyme unzips, if you imagine, unzips the DNA or the parent DNA at the replication fork. So that's just the name for this step. It's not actually a specific place or whatever, it's just the name for where this happens. So the enzyme unzips it, and then you end up with two sides. If you see how we got that, we've got these two. Basically what happens is it goes in and the helicase enzyme goes along and unzips it as it goes. Next, what happens is an RNA primase attaches the first few, I'm going to write this down here. Primase attaches the first few strands. Now I remember primase as like primary school. You go to primary school before you go to high school, I don't know. But yeah, the helicase enzyme unzips it, and then the primase, we're gonna choose orange. Okay, so the primase attaches the first few strands, so it just kind of gets it going. So we'll also make it look like a squiggle. I don't really know what it looks like, but yeah, this just attaches the first maybe 4 or whatever, and then a different enzyme. So we've got step 1, step 2, step 3 is a polymerase. Polymerase then attaches the rest of the DNA. So say if here we know that this is what the code looks like. The polymerase will then go, okay, obviously we knew that A attaches to T, so we can attach a T to all of them. Super easy. That makes sense, right? I'll just keep this over here so that we can remember this. The polymerase then attaches the rest of the DNA. We know this. However, we run into an issue because the polymerase only runs in a certain direction, which is where I think a lot of people are getting confused. On one side, this DNA runs 3' to 5'. It kind of goes this way. The reason why we name it this is because if we're looking at DNA, we'll go up here. The way it's structured is you've got a sugar, and then you've got a phosphate, that's my dog, and then you've got a sugar, and then you've got a phosphate, okay? And they're connected here. So say this is A, and then this is C, right? However, on the other side, so this runs this direction because this is the way the sugars go, okay? If that makes sense. However, on the other side of the DNA, it actually is the opposite. The sugar runs this direction. I can't draw, I'm so sorry, but the sugar runs this direction. See the difference? So this is facing this way, whereas on the other side it's facing this way. This is running from 3' to 5', whereas this is running from 5' to 3'. The sugar points to the 5' end. So the polymerase runs 5' to 3', which works great for this side because all it has to do is, we'll choose a color, we will choose blue. All the polymerase has to do on this side is just sit here and it'll just keep going this way because it's running 5' to 3' and it attaches as it goes. However, we run into an issue here because this side, as I said before, up here goes the opposite. This side runs 5' to 3'. The polymerase only runs 5 to 3. So if it was to go this way, it would reach the end pretty quick and it would run out, and then there'd be all of this on the other side that doesn't get done. So it's pretty clever. What happens? The primase attaches the first few, the same down here, attaches the first few, and the polymerase still goes 5 to 3. It then jumps backwards and it goes 5 to 3 and does a little section, then goes backwards and does 5 to 3. So it looks a bit like this, where it goes this, it does this section, then it comes back and it does this section, and then it comes back. So I'm gonna draw it without this just to try and reinforce this point. What it does is it does this bit, then jumps back, does this bit, and then jumps back. All right, so it goes this way, jumps back, this way, jumps back, this way, and then it keeps doing that until it's finished because it only runs 5' to 3', which I accidentally rubbed off. Okay, so I hope this makes sense. I'm going to rub this bit off to demonstrate to you now what happens. So, as I said before, these guys here are opposites. So where this is blue, this will be red, and vice versa. So, because of this, it means that if you have one side you can then have the other side. You know what the other side looks like, right? Because every time that there is red, we know that there is going to be a blue. So what this means is, with these two opposite strands of DNA, when the polymerase comes along and attaches the other side, right? Because the polymerase will go with blue. When the polymerase does this, it creates an identical strand as seen here. Notice how this is the same as this. And the opposite is also true. When the polymerase starts to do this bit, it does it, and it ends up with an identical DNA. Now, this is really important because now when the cell will divide, they will have the exact same DNA and be exactly the same. So yes, I hope this makes sense and I hope this is helpful. It's really important for you to remember that adenine goes to thymine and that cytosine goes to guanine. You only need to remember one pair, and I just try and remember that C and G are together and they just both kind of circles. That's how I remember. A and T kind of don't have a pattern in my head, but in my opinion, a C and a G are very similar letters. So that's how I remember that they pair up. Yeah, so I hope this is helpful.